This is podcast number 43. And one more reminder, your final is on Monday, the 7th, 12:15 to 2:15. You're going to take it online with Respondus like you have all of your tests. It's worth 25% of your course grade. It'll have 75 multiple choice questions, which works out to about 1.5 per podcast and recitation. Each of the multiple choice are worth 2.5 points. And then there's 9 extended response questions where you are going to pick 5. Those are each worth 12.5 points for a total of 250 points. And if I include something in this review, which is a very cursory review, doesn't mean anything in terms of being a clue as to it will or won't be on the test. Anything that was talked about this semester is fair game for the final. Okay, so continuing on our review, we talked about how eukaryotic transcription happens, how a cell decides that it will transcribe a gene or not. These are general transcription factors that are needed for virtually all genes that would be transcribed. In addition to these general transcription factors, there would be specific transcription factors for specific genes on top of all this. So before RNA polymerase gets to bind, there is a transcription factor 2D, and this is a transcription factor that serves RNA polymerase 2. RNA polymerase 2 is the one that codes or transcribes genes that are going to turn into proteins generally. And then there's the transcription factor binding protein here, TBP. Then transcription factors 2A and 2B join the complex. And then after that, you have RNA polymerase 2 with transcription factor 2F joining the complex. Notice that all these proteins have to lay down before RNA polymerase arrives at the promoter. Then transcription factors 2E and H join. H is the one that has helicase activity and also kinase activity. That kinase activity is important for phosphorylating the C-terminal domain tail of the RNA polymerase, which is important for the RNA polymerase leaving the promoter and actually finding the start site and transcribing genes. We talked about how in eukaryotic mRNAs there are introns that are cut out and exons that are spliced together. These will be processed by the spliceosome, and some eukaryotic mRNAs will be self-splicing, but in the spliceosome you have these small nuclear ribonuclear proteins. So these are protein RNA complexes that guide the spliceosome through its paces. You have an snRNP or SNRP U1 that binds to a consensus GU site on the 5' site of an intron, and a SNRP U2 that binds to a consensus A site, which is 5' of the 3' border of an intron. U1 and U2 interact, bringing the two parts of the intron together in a loop. They're joined by U4/U6 and U5 other SNRPs. Then there's a transfer of, there's a cutting on the 5' end of the intron and a transfer over to the A on the 3' end of the intron, forming this lariat structure. And then finally, the consensus AG sequence on the 3' end of the intron is cut. And the intron that is excised from the mRNA is degraded, and then there's a exon joining complex that brings together exon 1 and exon 2 with ligase-like activity. Here's an H5P question for you. We talked about how chromosomes move in cell replication, how that depends on kinetochore microtubules, which are different than other types of microtubules in the microtubule spindle. They are part of a kinetochore complex that has an outer layer and an inner layer that are proteinaceous and connect at the centromere of a chromosome. We talked about how cells progress through the cell cycle being the whole cycle of a cell's life, including growth phases, synthesis phases where the DNA is duplicated, a second growth phase, and then mitotic phase where the cell duplicates. These phase transitions through the lifetime of the cell are driven by the activity of cyclin-dependent kinases. Cyclin-dependent kinases are phosphorylating enzymes that depend on their activity depends on the presence of a cyclin binding to the kinase. The cyclin is destroyed, the kinase activity drops. These kinases can activate other complexes such as the anaphase promoting complex, which will push the cell through different parts of the cell cycle. One particular example of the anaphase promoting complex, it is activated by a kinase. It is a ubiquitin ligase in that it will attach ubiquitins onto things. It attaches ubiquitins onto securin, which causes the securin to be degraded by the proteasome. That releases separase. Separase is a protease that binds cohesins, which holds together sister chromatids. When those sister chromatids are no longer held together, they become separate chromosomes during anaphase, hence the name anaphase promoting complex. We talked about how there are checkpoints. One of those checkpoints has P53 as an important governor of that checkpoint. P53 is a very common oncogene, normally found in very low concentration because it's normally polyubiquitinated and destroyed. However, activity that is stimulated by damaged DNA can activate kinases that will phosphorylate P53, which will stabilize it, causing it to build up in concentration, and then allowing P53 to bind to DNA. One of two things can happen. It can recruit DNA repair mechanisms and cause cell cycle arrest so that the DNA repair mechanisms have a chance to fix damage. If that it doesn't work and that the damage is too extensive, it can recruit apoptosis mechanisms and kill off the cell so it doesn't replicate. We talked about how during meiosis in prophase 1 there is synapsis which occurs, which is homologous chromosomes lining up on top of each other and exchanging genetic material. This is one of the driving forces in the evolution of meiosis because it provides the adaptive advantage of variation in offspring. This prophase 1 is split into 5 different subphases that from the chromosomes first condensing to crossing over occurring, and then finally the nuclear envelope fragmenting. And we talked about how cells have a default mechanism to activate a process called apoptosis, which is a programmed cell death. So this is a different diagram than the one that's in your book. This is from a different book, and I think it's more helpful. Some people got confused. So your book is not trying to give you all the detail possible, because there is more detail, and this is not a senior level course. It's a 300 level course, not a 400 level course. But I think in that sense it may be failed. So I'm going to give you a little bit more detail here, and then just ask you to remember the level of detail that we got when we went over this for the first time. But hopefully this will be less confusing. Okay. So there is a growth factor, a trophic factor receptor that normally in a healthy cell will receive growth factor in the blood. If that happens, it activates a kinase. That kinase phosphorylates a protein called BAD. When BAD is phosphorylated, it's bound up by a protein called 1433, and then BAD stops. So the cell does not go through programmed cell death. Everything is good. If the trophic factor is not received, then the kinase is not activated. BAD is not phosphorylated, and then BAD inhibits an inhibitor. So this normally, there's an inhibitor preventing this channel from forming, okay, over here. So this is one unit of the channel, and this is all the units forming. Normally this light green one is inhibiting this guy from forming a channel, and BAD inhibits that. So BAD is inhibiting an inhibitor, which it can do if it doesn't receive the growth factor. And if this guy is inhibited, this guy over here is no longer inhibited from forming a channel. When that channel forms in the outer mitochondrial membrane, cytochrome C, which is part of the electron transport chain, is released. And then cytochrome C becomes a activator of a caspase cascade. So caspases are proteases that will chop up proteins in the cell and eventually cause the death of the cell. And one protease activates another protease, and so on. And eventually the cell will die. Okay, so again, in the level of detail that we talked about in the first time, if a cell receives a trophic factor, a kinase gets activated, and a protein gets phosphorylated. I don't think we talked about BAD necessarily. Can't remember. And that protein getting phosphorylated means everybody's okay. That's the end of the line. If the trophic factor is not received, so the cell is not getting a signal to grow, instead this inhibitor of an inhibitor allows cytochrome C to be released from the mitochondrion. Cytochrome C being released from the mitochondrion is a signal to activate a whole bunch of proteases, which will eventually kill the cell. We also talked about the involvement of PUMA if P53 is activated. So PUMA is another way where this inhibitor of can act like an inhibitor of an inhibitor and allow this channel to form, okay, through P53 and PUMA. And then we did not talk about PIM, and I will not talk about BIM here. Okay, so I just think this diagram is easier to understand, so that's why I included it in this review. Okay, after this we talked about specialized cells. We talked about neurons, we talked about muscle, we talked about HeLa cells. So you want to review that. That was not too long ago. You want to review that sort of thing. And of course you want to review each of the recitations and think of, what could be a fair multiple choice question, not too easy, not too hard, from these recitations. Good luck. You have now made it to the end of the podcast in cell and molecular biology. Congratulations.